Yeah. Assalamualaikum and very good morning, IB to all, especially to our guest speaker and the IMB staff, and also to our participants in today's webinar. So before we start our talk, we'd like to advise to all the participants to turn off your microphones so as to avoid any echoes or other audio disturbance throughout the webinar. Your cooperation is highly appreciated. So customarily to our webinars, I would like to make a really brief introduction to our guest speaker, Dr. Mathew Seymour from Hong Kong University. So Dr. Matt acquired his double degree, if I'm not mistaken, in wildlife management and also environmental natural resources from the University of Wyoming in the United States. So 4 years later, he acquired his master's in Holland University College in Iceland. And in 2014, Dr. Matt obtained his doctorate in the biodiversity of rivers and also river-like networks in Zurich, Switzerland. Currently, Dr. Matt is a molecular ecologist who is interested in using molecular tools to address various questions in biomonitoring and also biodiversity dynamics. So Dr. Matt has worked across a wide range of habitats. Those include freshwater, coastal, marine, and also terrestrial environments. His work aims to understand the drivers of biodiversity in natural communities and also the impact our rapidly changing world has on community stability and also ecosystem health. Dr. Matt's work also addresses how these effects can relate to the human society in general. So without further ado, I would like to invite Dr. Matt to start his presentation. The floor is yours.
Oh, thanks, Roman. Yeah, just share my screen here. All right, I think that works. Okay, yeah, so thanks. Thank you very much for the introduction, and thank you for inviting me to give a talk for you today. And so this talk will be essentially on eDNA and what it is and how I use it. And there's a lot of applications, and I'm happy to talk more afterwards, but this talk's going to be a bit more focused on biomonitoring and biodiversity assessment applications of eDNA. There we go. And so just a quick background on what biomonitoring is. And so when you want to assess like the quality of a given system, there's two main means to go about this, using either chemical assessment or biological assessment. And biological assessment gives us a better indication of long-term pollution effects on a given environment. So you can check the condition of what organisms are in a river or a site, and you can see whether they are consistently the same species or if they're like being, if they have a disease or whatnot. So it's kind of like a health check means to assess different ecological sites or environmental sites. And typically when we want to use biological information to assess environmental condition, we use organisms that are sensitive to environmental change. And generally when I say environmental change, I mean like chemicals and physical processes that are associated with either pollution or agricultural or human use. Like if you put a lot of concrete in, you can change the chemical composition of a river, which will impact the ecosystem services that are used. So not to get carried away. And the main aim here is that we generally use three main groups of organisms to assess environmental conditions, and those being fish, macroinvertebrates, and diatoms. You also find people using bacteria or phytoplankton in different situations, but these are the three groups that I tend to work with And traditionally, they are assessed individually. And so because it's a lot of work even to just look at one of these groups at a time. It takes a lot of specialization and a lot of taxonomic expertise. And there's a lot of different variations in the methodologies used to capture and assess these different groups. And so this is some of the examples. So I work primarily in rivers. I've done work in a lot of other things, but my big background is in rivers. And so we use things like kick nets, which is what you have here on the left-hand side, electrofishing for catching fish, and diatom scraping. So you'll scrape rocks to get at the diatom and biofilm composition of a river. And these all take different methodologies, and they all take different post assessments to get at what's underneath these samples. And so here's an example of looking for macroinvertebrates, where you have to size sort them into major morphotaxonomic groups. And then later on assess, as many of you are also aware, when it comes to actually taxonomically identifying organisms, it can be quite intense, even if you stay within a certain species groups. And here we're working across many orders of organisms to try and pick out unique individuals. And so there's a lot of limitations with doing this traditionally. And we're again focusing on freshwater fish, invertebrates, and algae, particularly invertebrates and algae. There are many small organisms. They're oftentimes very cryptic species. We can't identify some of the key players that are actually linked to environmental condition, because it takes a lot of effort to differentiate key species that are linked to polluted sites, even now. But we do, and there's also seasonal patterns that you have to consider. Capturing a lot of these organisms is very dependent on capturing them at the larval stage, which they are not always at the larval stage. They do cycle. And so sometimes if you sample in the wrong time of the season, you'll get different results. Although this doesn't necessarily, anyways, yes, depending on the seasonal. And so, and then oftentimes using traditional sampling methods is limited based on the physical conditions of the site. So for example, if I use kicknet sampling to sample rivers, I'm a bit limited on what types of rivers I can sample from, because you need coarse material of a certain size, so rocks, boulders that are the right size to appropriately sample from the river. If you try a sample from a river that is heavily channelized, so if it's all been concrete, or if the rock structure is a very solid rock where there's not much substrate, or if it's completely muddy, it's very, very difficult to get an appropriate and a suitable kicknet sample or a suitable invertebrate sample from those environments. Likewise, if the water is too deep, or if it's moving too fast, it can be very dangerous to sample using traditional methods. So there are some limitations to using traditional taxonomic approaches to assess biomonitoring conditions. And so that's what leads us to this new field of environmental DNA. I say new, it's been around since about 2007 with single species detections. And it's been about 2014 or so with the multi-species assessments. And we'll get into those differentiations in a little bit. But what environmental DNA is DNA that's been isolated from environmental samples that does not directly sample the organisms that you're looking to identify. And environmental samples can be pretty much anything that you can imagine. Very typically, it means sampling from water, soil, air. It can also be sampled from a microbiome, for example, if you want to take like a gut content analysis, or if you want to take excrement like feces analyses, you can take swabs for different surfaces. It's really very much dependent on what you are looking to target in the environment and how you want to relate that to your study questions. Water is very easy. It's probably the most common one because it's probably the most homogeneous. It's the most well-mixed environmental sample you can take with direct implications to what the ecological findings will mean. And while there's still a lot of debate regarding the interpretation of eDNA, because as you can imagine, taking a sample that does not directly relate to the tissue or the animal that you're hoping to get at, and saying that you extract all the DNA is highly contentious, even now. There's a growing body of work that shows that eDNA detection actually does relate to the actual species that are inferred from the environment. However, there's quite a bit of room to improve on these and strengthen these arguments, and we'll talk about that a little bit here as well. But the main thing about this is that eDNA offers a very cost-effective, once it's optimized, non-invasive, and a standardized way to sample biodiversity information from an individual water sample. And I guess drawing back on the fact that there's so much diversity in the sampling methods that I mentioned earlier, with one water sample, I can get information on fish, diatoms, and macroinvertebrates, as well as if I want to look at bacteria or other groups of organisms. And it's all using one methodological sample and using a pretty standardized group of molecular tools to do so. And so we'll just walk you through a little bit on what the whole eDNA process essentially. And so we start, of course, from sampling. I'm going to describe water here, but you can adapt this quite readily to a wide range of different environmental sources. And so for water, I will generally collect, of course, bits of water. Here you have an example of someone taking a cup and filling it, and you, of course, want to use, you want to minimize contamination when you do these things. But basically, you'll collect your water and you will filter it, either using a pump, or you can suspend it directly from the water itself. But the thing is, what you'll end up doing is you'll condense the material that you collect down to a pellet, and then this basically gets extracted using various different extraction methods. You can essentially any sort of extraction that you do with standard DNA techniques, you can do for an eDNA extraction method. There's a lot of proprietary kits that have been modified to deal with eDNA samples. And once you have once you have your genetic DNA, once you have your DNA extracted from your sample, you'll have a lot of various types of DNA that source DNA in the sample. This is where it does differ quite a bit from traditionally, because if you extract from a single organism like a bee or a tick or a fish, you know exactly what that tissue is. With eDNA, you get all sorts of stuff in there. And so let's say, for example, we actually want to see this crayfish DNA. We want to see if there's any crayfish in our sample. And with eDNA, there's likely very little material. If this was from the crayfish directly, all these fragments would be black, for example. But with eDNA, you probably have like 1 or 2 fragments of material that you actually want to identify within your sample. And so how do we isolate that individual DNA fragment in our sample? We use basically PCR to identify, isolate, and amplify these fragments. And of course, there's primers that you would design to specifically identify individual species fragments, and you replicate them and you make them glow in the dark, essentially using fluorescent dyes. And once you've done this PCR product, so there's 2 ways essentially currently that are most common to interpret, to get data from eDNA samples from after the extraction to the actual interpreting whether how much of a species whether a species' presence or absent in a sample, or how many different species you have in a sample. And so we're going to talk about first about the single species. So if you have done this step where you've amplified if you've done a PCR, this essentially is qPCR. I apologize, it's a bit out of order here. But essentially what you do here is you amplify all of your target individuals, you put them with a dye, and that's essentially what you're doing here with qPCR. And so if you want a process that is very quick, you have a certain species that you want to identify in the environment, you'll use this process to design species-specific primers that will allow you to quantify the amount of genetic material that is linked to that specific species. And this is a process called qPCR. It's very similar to PCR, but it allows you to assess the quantification of your sample in real time. And you do this for 30 or 40 cycles. Every cycle, the machine, this qPCR machine, takes a picture and it basically quantifies the amount of material that you have in your sample. It's a very quick and very, very cost-effective means to do eDNA analyses. I've used it for many experiments, and it's also used a lot for conservation biology. And if you want to, and you can multiplex this for across multiple species to assess presence, absence, but also quantification of individual species if you optimize it for your given region or for your given experiment. And for example, here's like a basic result. We have a bunch of samples. Each line basically represents a unique sample that you end up quantifying via the qPCR process. And so these lines, these different colored lines, basically show you how much how strong the signal is in the sample. And you have some black lines here that basically say there was no amplification. So these are basically samples with no crayfish in them. Of course, this is all, if you're familiar with all of the COVID qPCR tests, this is essentially what happens here as well. You end up getting cutoffs for different, anyways, I'm distracting, I won't do that. But there's a lot of similarity with COVID assessments based on, as there is with this kind of assessment. Anyways, and so if we want to not just do one species though, what if your question actually is more linked to biodiversity, and you want to look at more complicated linkages of the communities within a given sample? You can use next generation sequencing or high throughput sequencing to do things like metabarcoding or genomics, or even transcriptomics if you want, but they just increase in price, basically, on what I said there. The most common method right now is metabarcoding, which is basically taking one sample and massively you don't actually know what's in this. This methodology allows you to massively sequence multiple sequences from multiple different organisms using a single sequencing run. And this is a MySeq. And this process right here that I'm showing via the graphics is based off of Illumina technology. The most popular platform I think there is at the moment is still the MySeq sequencing platform. It's probably the most cost-effective and lab-friendly method. It's still a good chunk of money, but it allows you to do essentially a full plate of samples, about 96 samples, and you get a pretty good coverage of your samples for a community-based assignment. And this is done through massive parallel sequencing, where basically you attach these. I'm not going to go into this. I don't have time to do this. But if you basically end up attaching DNA segments to a plate, and over time, basically nucleotides get washed in to the plate and attaches them each line. It attaches them individually, and then once these basically form clusters of unique genetic sequences. And these end up being essentially what are your unique amplifications, these amplified DNA fragments. So instead of doing this for one, species, you end up doing this for multiple species, where each of these clusters represents a unique species group, or essentially a unique sequence group. And like I said, you get millions of reads from this. So instead of looking at one individual species with the qPCR, with metabarcoding, you can essentially look at tens of thousands of species, depending on the barcodes that you end up using. And so you get data like this, where you'll get over 1 million individual lines of data, and each unique line will have a nucleotide read. And then you can use programs, bioinformatic programs, to compare across these sequences and then cluster them based on their amount of similarity between the clusters. And then and so because multi-species, the major difference between multi-species and single species analyses is being that with the single species qPCR method, you know the species that you're trying to target. So you can be quite assured whether that information is accurate or not, because it's designed specifically to address that individual species. With multi-species, there's a lot more legwork and analyses that you need to take into consideration. One, because you don't have an assurance that that target, that the data that you're going to get is actually going to be 100% matching what's in the real environment. And plus, the amount of data that you have to process requires some bioinformatics analyses to clean up and quality checking that needs to be done in order to make sure the data is actually usable at the end of the day. It's the nice thing about multi-species is that you get a much richer data set and a much more complicated data set that you can ask a lot more questions in relation to biodiversity and community trophic interactions, et cetera. There's just a lot more. You have to be a little more careful when you are processing these data. And so the basic pipeline is something like this, where you take an environmental sample, you do an extraction, you run the PCR on the next-gen sequencing apparatus. Simultaneously, you also need to make sure that you have a reference data set that is based on taxonomic information and sequence barcodes that will then be combined with your sequence data in your bioinformatics pipeline to then give you the actual composition of your data, the taxonomic assignment information. And these reference databases are extremely important. And then they're actually the reason that we're able to do this type of analyses. And there are quite a few reference databases out there, from international collaborations to individual people that have created their own, sometimes hiding them away in their lab to be used individually. But the big ones are like NCBI, which is it's a wonderful resource. It can also be a minefield to go into if you're not careful. The BOLD system, which is primarily CO1 sequences, largely dominated by insects. Silva database, which has a lot of 18S, 16S sequences, which is a bit more universal in regards to primers. There's also things like Green Gene database, which is a bacteria-based one. And there's also some increasing ones for MyFish, which is a 12S fish primer. There's a growing number of databases out there. This is just a sample, but you rely on these existing databases to link the sequences that you get from your metabarcoding data to the actual taxonomic, the actual species in there. And this is a good paper by Buckle et al. And it really it kind of hits on the point that in the end of the day, the researcher is the person doing the research is really ultimately responsible for checking the quality of the data that you are end up publishing. And this is really important for multi-species reporting, because it happens all the time where it's very easy to generate the data, and it's just so much data that you end up getting. It's really important to also verify that the data that you are reporting actually makes sense, and that there's not some sort of issue with either the primer or the bioinformatics step. There's so many steps in the process that you need to make sure, be very careful in making sure that each step is verified and assessed prior to reporting the findings and interpreting the results. For single species, it's less complicated, but there are still a set of guidelines that need to be followed. This is a really nice one by Goldberg et al. There's a couple of more recent ones as well, but there are established there are much more established protocols for setting up single species eDNA primers. There's a whole list of criteria in regards to how to establish specificity tests, how to there's something called limits of detection, basically saying how many cycles you can use before the primer is useful or not, and also relating to testing your primers for how they react to different environmental conditions. This is a great paper to start with, though. And then there are some general eDNA protocols or eDNA considerations that you need to take when you are doing any sort of eDNA work. Because this material is quite easy to contaminate, you're dealing with eDNA in general, unless you're working with bacteria, eDNA in general is very highly prone to being contaminated from other sources. So if I take an eDNA sample and in the field, and I bring it back to my lab, I don't want to put that sample anywhere near where I'm doing standard tissue extractions or PCR amplification, because it's extremely likely that any sort of laboratory practices where I am cutting up tissue around the same area that I am working with my eDNA sample, it will very much likely just contaminate it either through direct contact through using gloves or even through aerial exchange. I've had so many instances, especially with colleagues' labs, where they don't separate eDNA molecular work from traditional molecular work. Can everybody hear me?
Yes, yes.
Okay.
Yeah, I can hear you.
Yes, sorry. So it's very important to separate eDNA work from traditional work. And it's also very important to separate PCR rooms from areas where you have eDNA practices. There's also a lot of work on making sure that you test your primers for your actual study. And there's also very standard reporting practices that need to be done for all these different, for different eDNA results. And that stems from reporting qPCR methods development, multi-species sequencing parameters, and environmental conditions used to sample eDNA in general. And it's also very, very important for eDNA to include negative controls into your experimental design. For example, when I go out to sample eDNA from a given site, I always take with me a negative control. And this can be for example, for water, I will take out a autoclaved liter of water that serves as a negative control, and I will process that at the same point that I process my other samples. Positive controls are also nice, especially if they give a stronger link to being assurance that what you have is working, and they can range. They're a bit more tricky to make sure, because you want to basically use something that is not native to an area, while also and they're more important for the molecular side of things. It's basically indication that the molecular process is working. And so with eDNA being material that you collect from the environment, it's also very important to consider the ecology of eDNA and how that impacts the interpretations of your data. And so when I talk about the ecology of eDNA, it's basically saying that eDNA interacts with the environment, and that interaction can impact your the interpretation. So for example, eDNA that's once it's released into an environment will degrade over time. And that degradation can be impacted by several factors. And so, for example, the origin of eDNA, does the eDNA generate from a living organism through basically peeing, pooping, whatnot? This size, the size of eDNA particles from living organisms is going to differ from organisms that are basically decomposing, because you have bigger chunks coming off of dead bodies, for example, and that can impact the time frame of that signal. Also, the state of eDNA. Different organisms will shed eDNA at different sizes. We did this for one example with Daphnia magna at one point, whereas a lot of other studies have worked on fish. Fish and smaller crustaceans basically shed eDNA at comparable rates, but they also shed at different sizes. Also, the fate of eDNA will differ between species in different environments. This can be due to either the population size of an organism, or it can be due to the actual environmental condition of the given site. We found acidity to be a key factor in one of our papers. Transport of eDNA will also differ across different environments. So for example, rivers will actively, of course, push eDNA down the river, whereas lakes, it can basically accumulate in the bottom of the lake and basically serve as a potential resourcing of eDNA if it gets if the sediment is disturbed, basically resuspending eDNA that was previously stored into the soil sediment. So it's not that it's just things that you need to be considerate of when you take eDNA from different environmental samples and different ecosystems. And then, well, so, you know, you actually want to use eDNA for, let's say, for biomonitoring, for example, here. And so applying eDNA findings for like regulatory means. And this is one example of how eDNA has been used to basically design a rubric for how we utilize eDNA in a government-based, like gosh, and then my brain just farts, in a government-based protocol, for example. And so for eDNA still is not going to replace traditional taxonomic approaches, but it can be used to supplement existing protocols because it is much faster. You can do a lot. You can do it much more rapidly. You can get results much quicker than taxonomic based methods. And so this protocol rubric was set up to basically show how we can incorporate eDNA into current protocol designs. And so if we have a site and we want to use eDNA, we can basically rapidly use eDNA to assess whether this was based off of using it to look at invasive species monitoring. And so if we want to start monitoring sites to make sure that we can start assessing whether a species has moved into an area, we can just use eDNA to blanket across a much wider spot than we can with individual species taxonomic surveys. And so based off how strong the eDNA signal is and the likelihood of the individual, so the y-axis is based on eDNA evidence, and the x-axis is based off the ecological understanding of the species. And so you can use the combination of eDNA detection strength versus the known ecological behavior of the organism to design basically a assessment criteria, or basically a protocol for how you would react given a certain situation. And then using And so that's a bit more traditional approach to if you want to combine eDNA with traditional methodologies. If we want to look at a little more forward thinking, there's a couple of things that eDNA science is currently undergoing in that regards, looking at improved biodiversity metrics and biomonitoring metrics, and also using eDNA to get at a more holistic approach to community diversity using networks. And the first one is a bit more traditional, and this is just an example of where we did a study a couple years back, and we compared eDNA traditional kickneck samples with eDNA water samples across a single catchment in UK. And we did this seasonally to look at how seasonal changes affected these indications. So taxonomic versus metabarcoding. And here we looked at species level information and also functional, and here functional being feeding group. It gives you indication of like fine particulate matter, and that indicates like whether something is agriculturally contaminated or not. And the main thing here being that we found higher eDNA biodiversity, which is what indicated in the blue colors here. The orange ones indicate traditional individual taxonomic identification. So we always found higher biodiversity using eDNA, and this is largely due to eDNA identifying a group of individuals that is not traditionally identified taxonomically. Chironomids, they're basically non-biting midges. They're very important for detecting differences in pollution and environmental condition, but they're very rarely used for environmental assessment. But eDNA is much better at picking them up versus trying to identify them taxonomically. We do find much stronger correlation between eDNA and traditional taxonomy when we look at groups that are traditionally used for ecological assessment, which is what these EPT richness indicates right here on the third graph set. EPT are a group of organisms that are very much easier to identify, and they have a longer history of being identified taxonomically. And we get nice correlation between the two, which is very promising for eDNA. And then overall, from all this information, the thing was that we get similar trends in eDNA versus traditional methodologies, but you get a much finer scale indication of what is happening in the system using eDNA, and you can separate out different effects using eDNA that you cannot otherwise do using traditional methods. And so traditionally, when we want to do biomonitoring, you usually use one set of organisms to assess what they're called biological quality elements. And you can combine them, and you can combine information from two sets to compare them across different sites, but you generally don't combine multiple taxonomic groups to get at an individual environmental measure. But we tried to do this with another study where we took a country, Wales-wide, which is Wales in the UK. We did across the whole county of Wales or country of Wales, and compared eDNA and vertebrate samples. We also using diatoms, macroinvertebrates, and we did fish as well, but the fish were a bit useless here. And the main thing was here that we got high correlation between using traditional metrics and eDNA metrics. This is a metric called average species per taxa, which is a standard one used to assess the environmental associated link between the organism, the site, and the environmental condition. Higher average species per taxa indicates a healthier environment, a less modified, less polluted environment, whereas a lower ASPT indicates a more degraded or polluted environment. And generally, these sites were not so bad. We don't have a lot of low ASPT ASPT sites, which is a bit of a downer, but we do get a bit of a correlation going on here. And there's a strong correlation between the ASPT generated from eDNA and the one generated from traditional methods. So that's a promising aspect. So that we had this, and these ASPT measures are based off of a lot of research from previous efforts looking at how individual species behave across different environmental conditions. And especially with diatoms, there's been a lot of effort to move away from traditional methods because diatoms are notoriously hard to identify in a very rapid effort. And so there's been a huge amount of effort in actually moving diatom biomonitoring towards to a molecular-based approach. And these are just some of the nice papers that have been recently published in that regards. Of course, moving from a single level approach of assessing, like moving from a strict community-based metric to one that actually takes into account the interactions between species is a very important aspect for further understanding the condition of a given site. With current bioinformatics understanding, we don't really look at when looking at individual sites, we generally don't look at the interactions between species. And unfortunately, this leaves out a lot of information regarding how the biotic interactions are impacting that site. If we just look at the current biomonitoring methods, biomonitoring interpretations, we're looking at just a purely abiotic effect on the community. We want to also look at the interactions between individuals within that community to pull apart the differences between the environment and the effects of the interactions on the given community to get a better understanding of actually what is going on in that site. And we can do this with using networks that combine the interactions between species and also the effects of the environment. So very quickly, this is an interaction. This is what we also did in this paper was we looked at the network interactions between species across different taxonomies and across the environmental gradient. I don't have a lot of time to go into this, and I feel like I'm just a bit meandering a bit, and I apologize. But very briefly, this is an example of a network of the total community across all sites. And if we isolate, and then each of these colors represents the environmental gradient associated across all of Wales. If we isolate individual sites, we can highlight here, for example, these are all the green sites. All these green dots represent one site within the total network, within the total point of Wales. And here you can see that they're all quite closely linked to an associated environmental condition. Whereas this site has individuals that represent a much broader set of environmental preferences. And the bottom one represents a site that has a lot more species that are somewhat limited in their environmental preference. And using this, you can extract network properties based on whether the group is highly connected, whether they are highly related. And this gives you an indication of whether and then you can relate those network properties to how healthy a site is, specifically based off of the biotic and environmental interactions. This was kind of exciting to work on, and I apologize, I'm probably not doing justice. I never do justice to this very well, but there's a paper, and you're more than welcome to read it. I feel like I write better than I talk usually. But the main results from this paper were that the criteria for designing networks will impact the results, indicating that we need justification for how we construct these networks. But we did a sensitivity test for this to basically highlight this aspect. And that the network properties that summarize community complexity are most indicative of the site environmental condition. And just very briefly, there are so many things you can do eDNA based analyses for. This is a really cool paper from Carl et al. 2018, and he did eDNA looking at river. These are a really nice group of modelers from Lausanne and Switzerland. And they basically modeled network properties based on eDNA data. There's been some really cool work from Thomsen and Sigurdsson in Denmark, where they took, this is more terrestrial, where basically they took eDNA from flower heads to get at what pollinators are impacting a given site. There's also some very cool work. I wish I pulled it out, but there's some very cool work also from Denmark, from another group, Bowman, where she designed 3D printed fans to collect aerial eDNA from zoos and also from nature parks, and found some good correlation. We've done some work with also taking galls and extracting parasites from, parasites and also parasites of parasites from taking galls from leaves. And so eDNA in general is quite widely used, and it's becoming increasingly used. There's a lot of course, pitfalls and traps that can be incorporated. I hope this gives you at least an overview of some of the stuff that can be done and some of the applications and research that have been applied. And of course, I'm happy to answer any questions if there's any. Thank you for your time.
Okay. Thank you, Dr. Matt, for your insightful and also very informative presentation. So I'm sure all of the audience, including me, can learn something, I think, about or two from your works. So right now we have come to the Q&A session. So if you have any question that you want to ask Dr. Matt directly, you can do so by turning on your microphones. If for those who don't have and unable to use your microphone, you can just type your questions in the chat box, and I will try to address them to Dr. Matt. So, any questions from the audience?
Yes, Raman. I would like to ask a question. Can you hear me?
Yeah, I can hear.
Yes. Thanks, Dr. Seymour, for accepting our invitation and for your great presentation. In fact, I'd like to ask one question to you. The absence of DNA in the sample doesn't mean the absence of the organism in the real environment. So in this situation, how do you deal with this situation?
Well, that's a very good point. And it will depend on what stage of the design you're in. One, if you're very confident that the species should be detected, it's probably due to a methodological something that needs to be rethought about in the methodology, whether with the primers you're using or with the sampling method that's being used. If you're not sure, if you're curious about something, you probably need to first test the method works on a controlled situation, either in the lab or in a semi-natural environment. So yeah, that's a very common issue, and it needs to be validated that the method works before trying to use it in a real situation.
Okay, thank you for your answer. I do have another question, if you don't mind. I think you show a very impressive network analysis, like showing the interaction between the organisms. My question is, can network analysis show the intensity of the interaction? For example, more frequent interaction, they will show a thicker line or thicker bar, something like that?
Yep. Yeah, definitely. Yeah, you can definitely add that as part of the analyses. Yeah. Yeah.
Right. Thank you.
Okay, so we have one question from Raj. in the chat box. He's asking about what about sequencing depth? What are the best practices to determine sequencing depth?
For multi-species, I think I generally you want to make sure that you have enough sequences. In general, you want to reach saturation of your genetic sequences for your given site. The sequencing depth is going to depend a little bit on your taxonomic focus. So if I have a set of species, if I have a community or a set of species that are diversity low, for example, fish, you don't need a huge amount of depth in order to make sure that you capture all of the fish in a given sample. If you're working on invertebrates, or if you're working especially on bacteria or plankton, where it's a huge amount of diversity, especially the more sites you add to a given study, or if you want to study across a very small regional area, you may not need a whole lot more depth. But if you sample increasing amount of area, you need to make sure you have a lot of depth in your sample to make sure that you have enough that you capture you fully capture the diversity in that sample. Probably just give you a little bit of numbers, perhaps. With invertebrates with macroinvertebrates in whales, for example, we aimed for about 30,000 reads per sample, and that was a sufficient number of sequences that we needed to ensure that we captured diversity across each of the sites.
Okay, thank you, Dr. Matt. Here's another question from Cecilia. Hi, Dr. Mathew. Thanks for the interesting talk. You mentioned that there's a risk of contamination during sampling and processing the samples. What do you say about sampling environmental water and bring it back to the lab to filter or preserve? How many negative control did you use without the process?
I tried to do as much of the processing on the site that I sample from as possible. And for water, for example, that means you can take water back into the lab and process this. That's been done before, and you can control that. I generally try to do that in the field, where that means filtering the water on the site and taking the filter back with me to analyze that in the lab. Regarding the negative controls, I tried to do one negative control per site. Depending on, of course, how your design is set up, some people do less. I'm just overly cautious when it comes to negative controls, because I use the negative controls. Negative controls are useful for screening out general contamination, but also site contamination. You can, if you want to process things in the lab, you just got to be extra careful to make sure that you're working in an environment that's not going to have PCR products or tissue products get into the water sample before you do the extraction, before you filter the water, and then also make sure that nothing gets in the filter after the fact. So it's just another layer of concern.
So Cecilia's asking another question. So what do you do if you have a positive feed for your negative control?
It will depend a bit on what the positive reads are. There are a couple of ways you can go about it. One, you can say, yeah, the whole sample. I mean, very, very, very conservatively is saying, yeah, the whole sample's busted. It's not going to work. If it's minor control, if it's minor contamination, and sometimes you can't. It's very hard to avoid. Sometimes, for example, bacterial contamination. Or human contamination. Human contamination is extremely common as well. You can basically wipe those from your analyses, for example. So anything that's detected in the negative control, you can basically you take out of the other all the other data that's related to that individual sequence. This can be detrimental sometimes, depending on what is in the negative control. So if you're working with fish and you get a species that you are interested in popping up in your naked control, it's a high likelihood that you may have contaminated all the samples with that particular either eDNA sample or something from the lab got in there, so that makes it very unreliable across the samples.
So Dr. Chin's asking another question. What is the best way to preserve the eDNA when transporting them back to the lab from the field?
Yeah, it depends on the sample. Of course, it depends on the sample source. Of course, for water, the best I found is to filter it in the field, and then that filter, depending on how far you are from the lab, you can either preserve it in things like ethanol or a lysis buffer, or if you are close by, you can also just freeze it. The main thing for the filter is to make sure that you don't have any water in the filter when you're transporting it. You basically dry pump it out. If you're doing soil or potentially air as well, you probably, there's a few things you can do. You can use liquid nitrogen to flash freeze it in the field, or you can use desiccant beads to make sure that the sample stays dry. Moisture is kind of the negative thing of dealing with eDNA. You don't want it will help degrade things faster. Heat and moisture is not really good for transporting eDNA. And what is your best? Well, it depends on how long you're talking. -20 is fine for quite a while. -80 for very long term is better. But it depends on if you use and this will depend on the preservative you use. So if you're using something like a lysis buffer or a ethanol to preserve your sample, you're not going to want to freeze them so much. You'll want to extract them, but they'll be stable in that form for, I think, up to 3 months. The lysis buffer especially is good for like 3 months, I think, at room temperature. But ideally, you get that extracted. Once it's extracted, it's quite stable. It's just prone to contamination if you're playing with them too much.
Jayavaj is asking another question. Have you tested RNA later to preserve generic material for this purpose?
Yes, yeah, and that works as well. Yeah. And then you can do, if you want, then you can also do the RNA from those samples for that as well. So yeah, that works as well.
So any other question to Dr. Matt? Okay. If there's no question, I think we can conclude our Q&A session. So the time is now 12:08. The time, unfortunately, the time is not on our side. So thank you, Dr. Matt, for being our guest speaker and also for sharing your research works. I'm sure we can all learn from your studies. And also thank you to the IMMB staff for organizing the webinar, this webinar. And also thank you, last but not least, thank you to the audience and also participants for spending your free time to attend today's webinar. So also last announcement, you can fill in your attendance in the link provided in the chat box. So without further ado, I can conclude our today's webinar for today. So next Wednesday we have another webinar organized by IMMB. So the title is still to be determined. We will inform you to WhatsApp and also email. So last but not least, thank you for attending this webinar. And so I bid you goodbye.
Okay, thanks. Thank you, Dr. Seymour. Yeah, thank you so much for your sharing. Yeah, and thanks to all participants for joining our webinar today. Thank you so much. Bye, everyone.